All right, good morning, everybody. Since we've invited a broader audience to this somewhat special ImmunoX Journal Club, I thought that a couple of introductions and explanations were in order. My name is Adrian Bakar. I'm a PI in the Department of Laboratory Medicine, and I organize this ImmunoX Journal Club. So this is a club where every week there's an alternation between students, or semi-alternation between students and PIs who present papers from the recent immunology literature. But it just so happens that Dr. Clifford Lowell, who's chair of laboratory medicine, was up this week, and he chose a very topical topic, COVID-19, and we figured we would advertise this a little bit more broadly because, as far as I know, there hasn't been a seminar yet on the basic biology of COVID-19, which is what Cliff decided to talk about. So for those who have not joined this seminar series before, welcome, and we hope that you'll come back in the future. It's every Thursday morning at 9 a.m. So just so everybody knows, I've muted everybody. I'm going to mute myself in a second, and at the end of the talk, of course, there'll be question and answer, and we're going to use that by using the hand raising button on the participants sidebar, and I'll remind everybody how to do that at the end. So without further ado, here is Cliff.
Okay, great. All right, Adrian, that looks okay, I hope. So thanks everybody for attending. Yeah, as Adrian just said, I'm gonna give kind of a broad, kind of medical school level talk on the basic virology, immunology, and pathology of SARS-CoV-2 and COVID-19. I'll start with this slide. This slide is a reference slide of all the currently available trackers and other interesting websites related to this disease. I'll actually go ahead and show you a couple of these. The first one is the Johns Hopkins website that you've all seen. Here it is. This is the one that's quoted every day and on NPR and all the radio programs that keeps track of infections worldwide. We can refresh it right there, and maybe somebody will want to write down that number, and we'll see whether or not it changes at the end of this talk. There's other trackers. This is another tracker, Worldometer tracker. This is an interesting tracker that is just been released. It was advertised in the New York Times. It allows you to zoom in by states and actually by counties, and you can see the infection rate. So for example, we can look at San Francisco. This is in fact San Francisco County. We can compare that to Los Angeles County, where they're having much more accelerated disease. So these are some example websites that everybody should know about, and they're all listed right there. Okay, so let's all get on the same page about coronaviruses. The virus is called SARS-CoV-2, and the disease is COVID-19. People frequently use these interchangeably. That's probably inappropriate. We really should refer to the virus as SARS-CoV-2 and the disease COVID-19. Coronaviruses are all single-stranded RNA viruses. They're envelope viruses. They have a plus strand. Their genome is about 30 kb. This makes them one of the larger RNA viruses in nature. And then this is the famous picture that everybody has seen of coronaviruses. They have this spike protein that lines the outside of the surface. The spike protein is the major antigenic determinant of the virus, and it's also the protein that engages the receptor, which I'll talk about here in just a minute. There are three groups of coronaviruses, alpha, beta, and gamma. Coronaviruses are widely present in nature. They infect birds and many mammalian species. We'll talk a lot about that in just a second. There are 4 established human coronaviruses. They're all beta coronaviruses. This is their names. And these viruses have been known since the 1960s because they cause many common colds and upper respiratory infections. It's estimated that their prevalence right now in the population is about 15%. So 15% of us listening to this talk probably have one of these viruses in us right now. And about 70% of people generally are infected. What this means, of course, which we'll talk about in a bit, is that we all have antibodies, endogenous antibodies to many of these coronaviruses. The 2 new pathogenic viruses that have appeared are the SARS-CoV virus, which gave rise to the first big SARS infection in 2002, which involves Singapore and Toronto. And then the second major pathogenic virus, which was the MERS virus that emerged in 2012 and involved the Middle East primarily. And now the current virus circulating is SARS-CoV-2. SARS originally infected about 8,000 people with a case fatality rate of approximately 10%. MERS infected only about 2,000 people, but had a much higher case fatality rate. The SARS virus has largely disappeared from the environment. There are no repeated or other outbreaks that have been described in the last 10 years. But the MERS virus is still around. There have been isolated cases and little outbreaks here and there, both in the Middle East and in Asia for the MERS virus. Okay. This is the genome organization of coronaviruses. This shows the genome organization of the current virus circulating, SARS-CoV-2, a bat-related coronavirus, and this is the SARS-CoV virus. And their genome structures are largely similar. The 5' ends contain 2 large open reading frames, all called open reading frame 1A and 1B. These encode large polyproteins that are cleaved up into about 16 small proteins that are involved in various aspects of viral replication, modulation of the host immune response. The structural proteins are all encoded at the 3' end here. There are 4 major structural proteins. The spike protein, that's obviously the one that makes it look spiky. The envelope protein, the membrane protein, and then a nucleocapsid protein. The spike protein is the one, as I said earlier, that binds the cellular receptor. The receptor is the angiotensin converting enzyme, or ACE2, and I'll say just a bit more about that in the next slide. Dispersed amongst these structural proteins are a number of other open reading frames that are named open reading frame 3 through 9. I don't know why these virologists, they can't come up with better names, but there you go. They call them open reading frame 3 through 9. And these open reading frames are much more divergent amongst coronaviruses and likely encode proteins that are involved in modifying the cellular innate immune response. And what we'll see, like all viruses, is that the coronavirus family is highly adapted to grow in the cells of its host by modifying the host responses. This is how coronaviruses gain entry into cells. They bind specifically the peptidase domain, the actual domain of the angiotensin converting enzyme 2, ACE2. And in that binding process shown here, both the spike protein and the ACE2 receptor get cleaved by cellular proteases. The major one is this one, TMPR SS2. There are other cellular proteases that can do that, that can cleave the spike protein at certain domains as well as cleaving the ACE receptor. And this results in a conformational change in the spike protein, allowing it to directly insert itself into the lipid bilayer of the membrane and facilitate viral entry. So this is, you can think of this like a cleavage of the spike protein and kind of almost a jackknife, a conformational change where spike then inserts directly into the membrane and allows the virus to enter. So blocking cleavage results in poor viral entry. Enhancing cleavage enhances viral entry. This mechanism is similar amongst a number of viruses. Influenza uses the same thing, this jackknife mechanism of inserting the envelope into the plasma membrane. The receptor for the virus, ACE2, is expressed on pulmonary epithelium and on vascular endothelium, primarily in the lung, in the heart, and in the kidney. And God gave us the angiotensin II enzyme to lower blood pressure. What it does is it cleaves Ang1 and Ang2 to smaller 7 or 9 peptide proteins or peptides, which oppose the function of angiotensin converting enzyme number 1, or ACE1, which actually produces Ang1 and Ang2. So these two proteins have opposite functions in nature to regulate both blood pressure as well as bronchoconstriction within the lung. Now, many clinicians, when they hear this, have asked, well, we know about ACE inhibitors. ACE inhibitors have been used forever to treat hypertension. They work by blocking the enzymatic activity of ACE. But I need to point out to my clinical colleagues that those inhibitors all work on the ACE1 receptor and do not affect ACE2. So the thought out there that these drugs may affect COVID pathology is wrong. They work on a different enzyme. But viral changes in ACE2, when the virus enters and ACE2 is cleaved, ACE2 levels are shed off the surface of the epithelium and in the endothelium, and that may contribute to lung pathology, in particular bronchoconstriction. Okay, so when I got into this, I wanted to know, why was SARS-CoV-2 so nasty? What made it different than SARS-CoV, and what made it different? What makes it different than other coronaviruses? And this has been worked out now by a number of groups very rapidly. In fact, most of all the data I'm going to describe was published like in the last 2 weeks. This is a paper actually from a group in Duke that compares the whole genome sequences of SARS-CoV-2 versus SARS-CoV versus a variety of other coronaviruses. And I have to acknowledge Satish Pillai out there. This was a group that he worked with, and Satish is my virology consult on this talk. But at any rate, if you look at SARS-CoV-2 and compare it to SARS-CoV, they're about 80% identical. The major differences being in open reading frame number 1 here, in the spike protein region here, and in ORF8, one of these genes that modulate the innate immune response. The majority of physiologic significance of these differences is really understood here in this spike protein area, which I'll show in the next slide. But what's really amazing when you look at this slide is, although SARS-CoV-2 and SARS-CoV, the original virus that caused SARS, are 80% identical, the current CoV-2 is 96% identical to this bat virus, right here in the little pink. You can see it all along here. And this is a bat virus. It's called TG13 that was isolated, I don't know, 12 years ago or something like that, a long time ago in China. And the one place where the divergence occurs between the bat virus and the current CoV-2 virus is just in this spike region. right here. And in fact, what occurs in this region is that the homology to another virus, a pangolin virus, increases dramatically. So bat viruses do not infect. This virus does not, the bat virus does not infect humans because its spike protein does not bind the envelope at all. If we look at this in another slide, this is a blow up of that. This is from a group also that published the same information in Nature Medicine. This is the whole genome. This is the spike protein. This is this receptor binding domain where all these specific amino acid differences are between the human CoV-2, the pangolin, and the bat. And what you can see is, if you looked at this carefully, is that in fact there are large sequences of, or specific sequences of the pangolin virus that have been substituted into the bat virus to produce the current human SARS-CoV-2 virus. So it's hypothesized that these substitutions result from a recombination between the bat and the pangolin viruses. That is, the virus that is infecting us is really a bat virus that acquired pangolin sequences. The other major difference in the spike protein is within a cleavage domain that separates these two subunits, as subunit 1 versus subunit 2. This is the cleavage that takes place that allows the subunits to insert directly into the membrane. The current SARS-CoV-2 is acquired from unclear origins a number of other amino acids in this domain that render the cleavage much more susceptible to a wider range of proteases. This is absent in both the bat virus and the pangolin virus. And this is called a novel furin cleavage site between the subunits. So these are the two major changes in the virus. That is, that this virus arose as a recombination between a bat and a pangolin, and also acquired furin cleavage sites that allow the spike protein to bind more efficiently. So the first thing I thought of is, what the heck is a pangolin? Okay, so what is a pangolin? So this is a picture of a pangolin from Science News, and I will read this to you. This is directly out of Wikipedia. Pangolins are used in Chinese traditional medicine for a variety of ailments, including excessive anxiety, hysterical crying in children, in women thought to be possessed by devils and ogres, and in malarial fever and deafness. So, you know, God knows. I don't know why it's not used in men that are possessed by devils and ogres, but so this is a pangolin, and, you know, whatever. But somehow these animals have coronaviruses, and it got together with a bat virus to create this recombinant. What is known is, and this has also been recently published, these are cryo-EM structures of comparing the CoV-1 spike receptor binding domain to the CoV-2 receptor binding domain. And in all cases, the substitutions that have been made by the pangolin viruses into the bat virus result in more contact sites between the spike domain and the angiotensin converting enzyme domain to facilitate or increase affinity. And it's estimated that the affinity of the SARS-CoV-2 spike compared to SARS is logarithmically higher, a log higher, in fact, maybe in the nanomolar range. So that's pretty high affinity. That plus this furin cleavage site may have facilitated the ability of SARS-CoV-2 to jump from bats into humans. So this is evolution maximus right here. I mean, this is as amazing as it gets. What is not known is what the effects are of these two other major changes in SARS-CoV-2 versus SARS-CoV. That is in the open reading frame 1 and open reading frame 8. And very little is known about these. It's thought that perhaps some of the changes in open reading frame number 1 happen to be one of the more divergent areas in coronaviruses, and this may also help facilitate rapid viral replication. ORF8, nobody really knows a lot about it or what it's doing, or at least I couldn't find out a lot about it. But it may be modifying or slowing the host development of an innate antiviral state. So these changes in the spike protein, these changes in ORF1 and ORF8 has resulted in a virus that binds to its receptor with much higher affinity, enters much more rapidly, and replicates really fast. And this was immediately apparent in the epidemic when people started measuring viral titers just by RT-PCR. This is from one of the first studies published in the New England Journal by a Chinese group, looking at the CT values for RT-PCR of SARS-CoV-2 and throat and nasal swabs from patients. And all of these viral titers are higher in the initial days of infection and start to drop with time. But you contrast that with the same sort of experiments that were done now back in 2003, 20 plus years, or 20 years ago almost, 17 years ago, where the peak of SARS-CoV replication occurs later, around 10 days, when patients are symptomatic. So for this virus, the titers are higher in patients when they are asymptomatic, or the days before symptom development. This has also been demonstrated in a smaller but a little bit more complete case study from 9 patients in Munich. All had known common exposure, so they could draw, they had, I guess they all met somebody that had a common exposure, so they could really get the days past symptom onset here pretty tight, measuring throat swabs and nasopharyngeal swabs or sputum or even stool. So the virus is detected in stool. It's important to know that the ACE2 receptor lines the gastric epithelium. There may be replication in the gastric epithelium as well, or frankly, this could be just virus that people are swallowing, they're expectorating from their lungs. But again, the viral titers are high early in an infection and then decrease with time. These measure just the log CT for RT-PCR reactions, but this group was able to convert it to an approximate viral titer. And the viral titer is around 10 to the 6th virions per I guess, mil of isolate. And this is similar to the viral titer of SARS as well. So it's not that this virus replicates to a higher level necessarily than SARS, but it certainly replicates more rapidly. This group also was able to correlate the approximate viral RNA levels with infectivity by measuring viral replication in tissue culture cells and doing classic culture. And in both throat swabs and nasal swabs, when the titer is the highest is when the culture is positive. And over time, even though viral RNA is detected in sputum and in stool, it is not culture positive. So it's not infective, probably. And really, you need a titer of virus around 10 to the 6th, at least as those virions are isolated from biologic samples, to show you a positive culture. These are the individual titers of all 9 patients in this small study, and they're plotted with the point in which these patients seroconvert, and I'll talk about antibodies in just a minute. And you can see that with seroconversion, the viral titers, kind of dwindle down, but not super fast. And in some people, they're kind of prolonged, and and so this is important to know. So that's what this emphasizes, that there's a slow drop in viral RNA post-seroconversion. What's also important to note of this study, and now other studies have demonstrated, that patients can shed the virus for as long as 22 days. So that can be as long as a week or 2 after seroconversion. Patients are still shedding virus. So the result of this is that SARS-CoV-2 is a highly replicative virus, but it causes lower pathogenicity. And this is from one of the websites on the first slide, this Information is Beautiful website, that plots on the y-axis the relative pathogenicity or percent who die, so that would be the case fatality rate, versus the number of people that are infected by each sick person. And you can see that if you take a really bad virus like Ebola or the flu virus that caused the 1918 pandemic, these viruses infect about 2 people. Each person passes it on to about 2 people, but they're highly pathogenic. Whereas the current SARS-CoV-2 that's causing COVID-19 is somewhere down around here. And you compare that to measles or chickenpox, which are rapidly spread, but are relatively less pathogenic. It is from these kind of studies that one can estimate what the incubation period is. And this is a plot of the incubation period. That is the time between when someone is exposed and when they develop symptoms. And that usually is on average around 5 days, but it can be as long as 12 days or as short as 2 days. And that's an estimation of the incubation period. So the fact now that SARS-CoV-2 replicates that it's adapted to replicate very rapidly and in patients in the respiratory epithelium means that it's easily transmitted by asymptomatic people. And it is these transmissions that you can estimate the R0, that's what was shown on the previous slide, of about 2.5. This is the R0 right here, an average number of people. There you go, R0. The fact that SARS-CoV-2 is transmitted by asymptomatic people has been demonstrated also in the study of these poor people on the cruise ships. I don't know why anybody would go on a cruise ship. Infectious disease doctors tell you not to go on cruise ships. The patients there that have been studied, that as many as 15% are asymptomatic. And this is a case study published again from a German group recently in the NEJ, where they had specific contact tracing to demonstrate the fact that the virus is transmitted by asymptomatic people. This occurred when an index patient, a woman from China, visited Germany for a series of meetings in January 19th, 20th, and 21st. These are 2 German businessmen that met with her. She was asymptomatic. She flew home back to China, developed symptoms a couple of days later, and actually had a positive PCR on the 26th. Both of these guys met with her. They developed symptoms. Now 4 or 5 days later, they turned positive. And this poor guy, patient number 1, transmitted the disease to patient number 3 and 4. These are the specific dates in which these people had contact. And they too developed symptoms later on. So it's this kind of contact tracing that allows people to calculate what the R0 is, or how many people one infected person can often transmit the virus to. So at the end of the day, SARS-CoV-2 is a highly adapted zoonotic virus. It's evolved from bats via recombination with pangolins and other animals. It's acquired a spike protein that allows it to insert rapidly and efficiently into host cells and bind its receptor with high affinity. It probably also works to modulate the host innate immune response. It replicates rapidly, but doesn't cause a lot of pathology compared to most other viruses. It's pretty stable. It's secreted in stool and may replicate in the GI tract. People probably know about this paper, also just recently reported in the NEJ from a group at the NIH that grew up SARS-CoV-2, and tested its stability on different surfaces to try and answer how long does it stay in the environment. And the half-life is about 6 hours on plastic and steel. So that is that if you put 10 to the 6 virions on these surfaces, you can detect the viral RNA at 10 to the 3 or 10 to the 2 levels days later on the surfaces. And that was borne out also, these case reports of detecting virus on the surfaces of the cruise ships that have returned as many as 22 days later. Whether that's infectious virus or just viral RNA is another issue, but it's a pretty stable virus. So the result is that nature has produced an infectious machine that is something to be awed at. You don't mess with natural selection. All these papers that have looked at the natural origin of the virus have pointed out, though, that this evolution is consistent with natural evolution. It is not consistent with some artificial construct in the laboratory. There's a lot of rumors that are out there and that this is a man-made virus. And everybody who has compared all the genome sequences of it with other related viruses have come to the conclusion that this is a product of natural evolution. Okay, so I got to thinking about this. You know, all these bad viruses are coming from bats. So what's the deal with bats? Why are bats producing SARS virus? The virus that caused SARS passed from bats to civet cats that gave rise. The MERS virus, Middle Eastern Respiratory Virus, came through camels. I just told you the virus that's infecting us came from is a recombinant between bats and pangolins. You know, what's the deal? Bats are also the source of Ebola viruses, Marburg, rabies, Hendra viruses. All these viruses in humans and us cause big infections, then sterilizing immunity. But in bats, these produce persistent low-grade infections. What's the deal with bats? So it turns out there is a whole world of bat immunology out there that I got into. And there's a variety of reasons that bats serve as the reservoir for many of these viruses. Many of the bats have very polymorphic receptors so that many of the viruses can barely infect. But the real reason bats are such reservoirs for these infections is that many of them have very robust innate immune, in particular type 1 interferon responses. And some bat species actually produce type 1 interferon constitutively and have constitutive expressions of interferon-inducible genes and express multiple interferons besides interferon alpha. And they compensate for this by often downregulating other innate receptors so that they can establish a constant state of viral tolerance or viral, a relative lack of viral tolerance. That's the best way to say it. And there are many people in the bat world that suspect that bats have done this, evolved this tolerance mechanism for reduced inflammatory responses, but a constitutive antiviral state to adapt to the metabolic stresses that occur in them when they fly around in caves. So this is great, but how does this translate to the evolution of human pathogens? And lo and behold, again, just in February, a wonderful paper came out from a group in Berkeley that looked at this. They submitted this paper back in May last year, so this had nothing to do with SARS-CoV-2. But they addressed why bats are the source of all these nasty viruses. And the experiments they did were to culture 3 cell lines, a Vero regular African green monkey cell line, and 2 bat cell lines, one of which constitutively produced alpha interferon, infected them with different viruses, and looked for rapid spread of the virus across the cultures. And what they found was that the bat cells were resistant to many types of viruses. And this is a wonderful paper. These are the primary data. The paper is full of all this math that they made all these models. Anytime there's a biology paper full of math, I'm both intimidated and think it must be really good. I don't know, who knows all this math? I'm just a biologist. but these are their models. But you can see if you look at the data, this is the Vero cells, and what happens is all these viruses replicate to a very high titer, kill all the cells in the culture, and that's the end of the virus. Whereas in this cell line that produces alpha interferon constitutively, the virus doesn't replicate very much and then just stays in the culture continuously here. And their mathematical models model that. And the basic answer is that the very strong antiviral immunity that bat cells have promotes very rapid viral replication in the early stages just to get persistent infection. So it's as if the strong antiviral immunity of bat cells turbocharges viral evolution. And that's actually Satish's word, not mine. Thank you, Satish. To evolve viruses rapidly. So strong antiviral immunity is great for bats. It keeps them from getting infected by all these viruses, but it promotes a lot of viral evolution and switching of viruses to human species, and that's bad for us. So that's what's going on with all the viruses. We can follow viral evolution in this pandemic by now all the genomes that have been sequenced through viruses, and all of this is on this wonderful website called nextstrain.org. I'm going to actually show you that website. So let me get out of here. Let me go to the website. Yeah, here it is. So this website compiles all the full genome sequences of all the different novel coronaviruses in different companies here or countries here in China, Europe, and in the United States. And by comparing all these, you can see the evolution of the viruses. So you can see that there are basically 1, 2, 3, 4, maybe 5 major clades of viruses that are out there. It can be displayed in a radial fashion here that maybe a little bit better. Here it is in radial fashion. Here it is in an unrooted fashion. Let's see if that plays. And here you can easily see how all the viruses have evolved from each other. Most of the viruses that have come to the United States came from this original founder virus. This was a virus that was transmitted by the first guy who came from China and went to the state of Washington. He infected about 4 or 5 people in the airport van coming back from the airport. But there have also been other viral isolates here, one in New York that comes from a different clade, and so on. And so if you display all this in a rectangular fashion, goes back to the rectangular fashion, you can actually on the right-hand side play this out and see the evolution as it's occurring. First here in China, and then here in China, right here, and then the virus is emerging out from China, and then you can see, boom, this first virus right there that hit the United States. That's this one right here going to Washington State. That's the guy in the van, and you can play them out, and you can see then that some went to Canada, and then Canada back to the United States, to Europe. Here they end up in Iraq. This is where the viruses in Iraq came from. Here's a virus from Iraq going to Australia, et cetera, et cetera. So you can spend a lot of time on this NextStrain site and follow viral evolution. Many of these sequences have been contributed by Charles Chu in our department in laboratory medicine. And he has a paper that's just come out on that as well. Demonstrating that a lot of the viruses that have infected us here in the Bay Area have come from multiple sites, mainly from China. Okay. All right, so this brings up testing. So how do we test for the virus? And there's some interesting stuff in here. Most of the tests for the virus are simple RT-PCR reactions. They amplify an RNA-dependent polymerase, the RNA-dependent RNA polymerase in one of the open reading frames, as well as unique domains in the envelope and the nucleic capsid. And these are standard RT-PCR reactions that were developed in China, now used worldwide. The primers may differ a little bit. They're basic TACMan. Everybody understands this. But there's new assays that have emerged, and it's like seeing how a crisis drives technologic development. And I think these are pretty cool. Some of them are isothermal RPA assays. So that, I'll bet you 90% of us on this call don't know what an isothermal RPA assay is. That's a recombinant polymerase amplification assay. That's just been commercialized. And then there's CRISPR-Cas12 and Cas13 nucleic acid detection assays. So I thought I would just tell you what an RPA assay is. It works just like a standard PCR reaction, but it depends upon different proteins, mainly from bacteriophages, recombinases, single-stranded binding proteins, and a strand displacing polymerase. The primers bind to the 3' and 5' ends of the target, and are actually introduced by the recombinase, and then the single strands are stabilized by the single strand binding protein here, and then you get amplification. And this all occurs at one temperature, so you don't need cycling of the reaction. This means it occurs much more rapidly, so you can get amplifications of the target sequence within minutes. And you can detect these amplifications either by standard TACMan probes like this probe, or by using probes that actually have biotin on it, so that you can end up detecting the amplicon, or the presence of the amplicon, with a little TLC lateral flow detection method. It's really amazing. And this is the technology behind this new test that every one of us has heard about, this 5-minute detection test from Abbott. I literally heard the governor talking about this this morning on the radio on NPR about this 5-minute COVID detection. And these are all RPA, isothermal RPA assays. I'll bet you nobody's heard about Cas12 either. We all know about Cas9. Cas12 is another cleavage enzyme that's driven by hybridization, comes from different organisms. It is different than Cas9 in that it binds its own guide RNA and detects its target and then cleaves across from the target. When it does so, it also activates a non-specific single-strand DNase activity. So all of a sudden, the enzyme will degrade single-stranded DNA anywhere in the environment. So this allows you to put together an assay where you have the Cas9 or the Cas12 and its guide RNA to whatever COVID sequence you want. Just bind it directly, then put in a probe with one of these biotin probes, and lo and behold, detect it on a little lateral flow strip. This is like a pregnancy test. You could do this you could do this in a test tube if it was really sensitive enough. And these assays are now all coming out. This actually was also done by Charles Chu's group here at UCSF. So I thought that was some interesting technology being driven by this epidemic. This brings me to serologic testing. There are many serologic tests that are coming on now. Most of them are targeting the spike protein. Most of these are ELISA type assays. Many of them currently are of suspicious validity, but they will get better soon. And we will have these in clinical use pretty soon. This is some studies, the first study from China showing that everybody develops antibody responses within 2 weeks after the onset of symptoms. This is for total antibody, IgM, and IgG, correlating it with viral RNA. I'm not sure in this experiment why IgG and IgM seem so much less than total antibody. You know, but that's one example of it. These are other examples here against individual patients developing antibody responses. This is a better controlled study from Europe, listed here, again, just published, showing correlation of IgG levels with the degree of infection. And demonstrating specificity, because only really people that are infected, this is serum samples now from people infected with SARS or MERS or other viruses, they don't react in this assay, whereas only serum from patient with SARS-CoV-2 or SARS, 2 of them react. What's really important about this study, though, is they also did ELISAs with all the other coronaviruses and look at how much antigen reactivity or antibody we all have to all the various other coronaviruses, whereas we don't have antibodies as much that are specific for MERS or SARS. So this raises a general question, which us immunologists would be interested in, is whether or not antibodies in general are protective. Is a vaccine really going to work? So we look to the past infections of SARS and MERS to guide us in this. And one thing is completely clear, and that is that therapeutic antibodies made, specific monoclonals made against S protein work great in various animal murine and macaque models for both SARS and MERS. And usually they work by blocking binding directly. There are some viral escapes in some of these models. Many of these studies talk about using 2 monoclonals at one time to avoid viral escape. Interestingly, Gary Nabel had a paper way back in 2005 showing that some of these monoclonal proteins actually activate the S protein. They do this unfolding and allow for infection. So not all monoclonals are bad or good, but we're going to see these types of monoclonals for therapy, for passive therapy, very rapidly emerging from a whole variety of companies. But this is not a vaccine. This is not this is passive immunity. To ask whether or not there's really adaptive immunity in this infection, you kind of look at correlative studies, and there are correlations of serosal IgA level with disease activity in patients with MERS. The problem is that antibody levels fall rapidly in patients post-recovery. And in fact, even in the SARS epidemic, people's Ig levels declined over time. So the real answer is we just don't know if you become immune post-infection. But we can look to vaccine trials in animals to see whether or not post-infection immunity or even vaccination will work. And this has been done widely in the SARS and the MERS epidemic prior to the current epidemic. And vaccines, both subunit and inactivated vaccines, work great in mouse and macaque models to produce wonderful antibody responses. But in many of these models, when the animals were re-challenged, they did worse. That is that the vaccination impaired immune responses. It made the animals more susceptible to infection, completely what you would not expect. And this, we now understand, is the phenomena of antibody-dependent enhancement of infection, or ADE. And this is a well-described phenomena for vaccines for dengue viruses. That is that you vaccinate with one type of dengue, and then patients get infected by a second type, and their outcome is worse. And in fact, for dengue virus, this ADE enhancement is actually thought to be the cause of severe hemorrhagic reactions to dengue. The mechanism by which this occurs is thought to be this: that is, with the first infection or the first immunization, you produce high titer, very reactive virus that results in viral neutralization or uptake into lysosomal components and macrophages. But with the second viral infection, the titers may be lower or partially cross-reactive, and instead you direct uptake to other internal vesicles in phagocytes that actually are not virolytic, that allow viral escape into the cytoplasm. And so all you're doing is enhancing the ability of the virus to get into cells, into non-lysosomal subcellular compartments. So many people are worried that this is exactly what's happening right now in the COVID-19 epidemic, that many people have antibodies to coronaviruses, that it may be cross-reactive, and that the reason a lot of people get such a bad outcome is that they're having an antibody-dependent enhancement of infection. And this is my personal hypothesis for why older people are more susceptible. I'll come talk about that in a minute. This has a lot to say about recovered plasma as a therapy. Oh boy, I don't know, we'll see. We're gonna have to be careful with that. Okay, but clearly there's a lot of effort on the vaccine side that we're gonna see. DNA-based vaccines, some are already in clinical trial. Changing the adjuvants can avoid antibody-dependent enhancement. It's clear that DNA-based vaccines or inactivated viral vaccines will induce cellular immunity. We know from animal models that cellular immunity is important in the response to coronaviruses. This has been demonstrated in transgenic mouse models for SARS. T-cell responses are important in MERS and macaques. So clearly this is the kind of viral vaccine rather than a classic subunit or boiled virus, inactivated virus that's going to emerge, though it will, as everybody says, take time. And to understand how to make an inactivated viral vaccine, you have to understand how the virus inhibits the innate immune response. And at the end of the day, it's really the innate immune response that's generating the pathology. And these viruses, like many viruses, are just an amazing, have an amazing repertoire of tools that turn off innate responses. And this is from a figure all talking about SARS and MERS, and presumably it's saying the same is true for SARS-CoV-2, that many of the non-structural proteins block type 1 responses. They act as phosphatases that dephosphorylate IRFs. They have, some of them block type 1 interferon responses. Some block proximal RNA sensing by the RIG-I mechanisms or MDA5. Many of them work at the transcriptional level to block productions of ISGs. The SARS M protein may even work up here to block IRACs in initial. So these viruses really work to turn off the innate immune response to the viral state. And it's when this becomes dysregulated, this innate immunity, that's when you get pathology. That the pathogenic or dysregulated inflammation allows for delayed type 1 responses, robust viral replication, leading to enhanced epithelial apoptosis, suboptimal T-cell responses, acute lung injury, and death. Whereas with a more protective response, especially with early interferon responses, you get a much more protective immunity and host survival. So at the end, this is just another way, another reference to portray that early infection resulting in widespread pulmonary epithelial apoptosis or pyroptosis leads to primary inflammatory responses that become dysregulated. Recruitment of innate cells, neutrophils, and monocytes. Neutrophils are known to play a major role in the pathogenicity of acute lung injury. And this results in immunopathology, and patients end up in the hospital and on ventilators. And this is what their lungs look like. It's a classic acute respiratory distress syndrome, ARDS picture, accompanied by a cytokine storm of elevated levels of all sorts of inflammatory cytokines. This involves both the lungs as well as the liver and even in the heart. And many of the patients that we're seeing now that that die of this disease die of a myopathy as much as they die of the lung disease. There's loss of lung barrier function, poor oxygen exchange. That's why patients have to go on ventilators, as well as significant other tissue injury. This is a classic picture from Michael Mathey. Michael, I hope you're on this talk. This is one of his pictures he shows in all his lectures of what the alveolus looks like in an injured state, full of neutrophils and alveolar macrophages, losing the surfactant layer, full of fluid, not exchanging oxygen. That's a shout out to our expert on ARDS here at UCSF. So this is the overall clinical course of what the infection looks like. These are seminal studies from the Chinese group early in the outbreak. That early in disease, you have patients with high viral shedding, modest symptoms, low dyspnea, but then they start getting short of breath. They go into the hospital, they get dyspnea. But in fact, this is when they're not shedding as much virus and they're suffering really from immunopathology. And in this slide, they clear virus. If the patients are non-survivors, they don't clear virus. They progress all the way to death. So this raises the question. This is a slide I completely stole from our infectious disease team about when to treat and what are the right therapies. And obviously, early in disease, when viral replication is high, that's when you want to do therapies that are antiviral in nature, blocking viral entry or blocking viral replication. But when the disease results in immunopathology, blocking the virus at this state, the horse is already out of the barn. And really what you're talking about is immunomodulatory therapies at this point. And I think this is the general thinking in the field. It's just a matter of what are the right antivirals early and what are the right immunomodulators late that we should be using. The final question, and I'm racing, I realize I'm over time, is why are old people so susceptible, so worse with COVID-19? I've got a personal interest in this since I'm in the risk category. One of the clear things is it's not just because children are not infected. In fact, children are infected. And this was demonstrated in a nice Chinese cohort study published also in the NEJ, where a random survey of 1,300 kids, about 12% are infected, but the majority of them had no symptoms and only one severe case. And this was a poor kid who already had leukemia. So it's not that children are not infected. They are infected. They just don't show symptoms as much. And this age issue, interestingly, is also reported in macaque monkeys. So this is a study with SARS. It was done now, 10 years ago, infecting a cohort of 6 young monkeys and 6 old monkeys with the SARS virus. And the poor old monkeys got much more fever. They had worse lung injury, more pathology. This is what their lungs look like over here, a lot of alveolar thickening and inflammation and respiratory shedding. And there was a lot of expression profiling in this paper. I won't go over all of it. But the authors traced all this to a weaker type 1 interferon response in the older monkeys. And in fact, if they treated some old monkeys soon after infection with recombinant interferon alpha, they could convert their pathology or convert their susceptibility to that of a young monkey. So what's different about the immune system of old people? Well, maybe it's, I think, or it could be, this constant exposure to pathogens, seeing coronaviruses all the time, having higher antibody titers that induce ADE, that may be it. It might be that older people have weaker innate responses. They're slower to mount a type 1 interferon response. There's some studies of this in humans. There's also a correlation with chronic inflammation. So it is true that older people have more states of chronic inflammation. This may modify their innate immune responses and adaptation to the chronic inflammation. This would all be post-transcriptional or post genomic. This would probably be, chromatin modification type stuff that would keep our innate responses in check to help limit the amount of chronic inflammation we have as we grow older. So this may be actually adaptive for older people, older monkeys, to have a weaker innate response. And there also could be changes in T cells and B cells. My final data slide on this is a paper that just came out, so I should say 2019 in PNAS, of a single cell RNA-seq experiment of peripheral blood mononuclear cells, comparing a cohort of Japanese people that are 100 years old versus some that are 50 years old. And here's the Tisney plots, and I'm just showing this because you can't have an immunology talk these days without a Tisney plot. So there's your Tisney plot. But the result is that the older people had fewer B cells, and they accumulated this weird cytotoxic type CD4 cell that was kind of oligoclonal in nature. So if these are directed towards coronavirus, you can imagine that would be trouble, or you can imagine that a more limited repertoire of T cells in older people may also contribute to their susceptibility to this virus. But this doesn't explain the age difference for COVID-2 or for SARS-CoV-2 or a COVID-19 disease compared to other viral pathogens. Influenza affects both the young and the old. This disease, it's the old. So the answer to this is, who knows? I don't know. I don't know why older people are young. And so there's your joke slide for this talk. I'm going to end by showing you some of the predictive tools that have come out for predicting the outcome of the epidemic. And you should be aware of these. This is one of them. This is from a group at the University of Washington. Let me refresh this. That have used a variety of predictive data when people were doing social distancing, the amount of available beds and such, to predict the outcome of the infection. So here's today. We're right there today on April 2nd. This shows for all of the United States, and you can see that they are predicting that the peak of all hospital beds that will be needed in the United States will occur in April 16th and then wane. And then you can go and look at this state by state. So if you look at some poor state like New York here, there's New York. You can see that they're predicting that the peak will occur very soon, April 19th, and will be above the number. This flat line across here is the number of hospital beds that they have. And you can actually look at the number of ICU beds, and they're predicting that the number of ICU beds needed is 12,000, and the number currently available, at least based on this data, was 718. This is some scary stuff. Let's look at us here in California. And if you look at all of California, what you can see is that the peak is way more right shifted, that we're predicted not to peak until later in the month of April and May. And when we do peak, if you look at all beds, we're clearly below the number of beds we have available. And if you look at ICU beds, we're just barely below the number of ICU beds. And I think this is what people are saying about flattening the curve. This is evidence of flattening the curve in California. Okay, so let me get back, and I will end my talk. We can take, I'm sorry for going over, we can take some questions. I want to say, I want to give a shout out that there's a medicine grand rounds that's occurring today at noon. This is the Zoom link for it. It will have much more clinical information. You won't be talking about bat immunology in this talk, but it'll say a lot more about therapy. And I think these two talks hopefully are complementary. So I'll end my talk there and turn it over to Adrian.
Great. Well, thanks, Cliff. That was a fantastic talk. So as Cliff mentioned, he's very happy to take questions. I've noticed that we had over 250 participants over the course of the last hour. So in order to try to keep this, no open mic, or else I think it'll be a disaster. And so what we'll do is have people raise their hands, and I'll call on them, and we'll see how that goes. And the way to do that, in case you don't know, is on the bottom of your screen, if you scroll down on that, you should see participants. You hit the participants button, and then on the right side, where the sidebar comes up on the lower, toward the bottom of that, it was for me on the right, is a raise hand button. So I'm seeing people raising their hands, which means it's going to work. So when I call on you, I think it's going to unmute you automatically. If you want to be seen, turn on your own video though. And my apologies if we keep pushing the buttons for the muting and unmuting. And one last thing is that we talked about this earlier, Cliff and I, that to try to hold these questions, since I'm guessing there's going to be a lot of them, to more basic biology and clinical course kind of questions rather than UCSF specific or Bay Area specific questions, because I think then we'll get a little bit overwhelmed. So, okay, so I'm going to call on Mark Anderson here. Hi, Mark.
Hey, good morning. Hey, Cliff, fantastic job. I really enjoyed it and really did the yeoman's task here. So on behalf of ImmunoX, I want to thank you for that. So to keep it brief, I think my question is another interesting comorbidity that you can imagine I'm constantly getting asked about is diabetes. And it is very impressive how diabetes looks like a comorbidity for a bad outcome. And I think it just like age, it really is an outlier. So what are your thoughts on that?
Your guess is as good as mine. It seems to me that a lot of this is really the speed with which we mount an antiviral response, a type 1 interferon response. Bats do it super fast, and it may be that we're just not mounting that as quickly. I don't know how type 1 interferon responses are affected in patients with diabetes. But maybe there's some correlation there. I just don't know. Of course, nobody knows. You have a thought? You're on mute, Mark. Okay, yeah, unmute him, Adrian.
Here we go.
All right.
Yeah, no, I mean, I think like, just like some really simple experiments that could get done right now is the antibody, do they mount a decent antibody response compared to normal patients?
Yeah,
right?
For sure those studies are going to be done because everybody's collecting serum from everybody. Will be able to correlate antibody responses with clinical outcome. Those data will be forthcoming.
Yeah.
Great, thanks, Mark. Arun.
Hey, Cliff, that was a fantastic up-to-date summary.
Hey, Arun.
For that. You mentioned, Cliff, that the ACE inhibitors don't affect ACE2, but indirectly, could they potentially increase the expression of them? That's what I'd heard, and maybe that increases infectivity.
Yes. So that is, I think I saw that too, that there is this reciprocal balance between ACE1 and ACE2. And when you give ACE1 inhibitors, what happens actually is that you may actually increase a little bit of ACE2. So there may be a reciprocal balance there. The same is true too, that when you shed ACE2, that there may be more activity of ACE1 and the two may kind of counteract each other. But the main thing is that that there is no direct effect, and that's what I wanted to point out.
Great, thanks.
Great, thanks. Jeroen, you have a question?
Sure. Cliff, so you showed the map of the basically the genetic evolution of the SARS-CoV-2 virus. Is there already data out there in terms of binding to ACE2 or different infection rates?
Well, there is the cryo-EM structures that I showed, and they do give a higher estimate of the affinity of SARS-CoV-2 spike protein compared to SARS-CoV, logarithmically higher, and they're estimating that it's a nanomolar binding affinity. And for those of us on the call, nanomolar is high. I mean, that's like biotin avidin. That's even higher than most antibodies recognize antigens. Tony
But
may
I,
correct me on that.
but I meant for the SARS-CoV-2 itself.
This is the spike protein in SARS-CoV-2 binding to the binding to ACE2.
Mhm.
That its affinity is clearly much higher than SARS-CoV. Ask me again, Jeroen. Ask me.
So are there already genetic polymorphisms in the spike protein within SARS-CoV-2?
Though the main polymorphisms already during the epidemic.
Right.
No. Most of the what I didn't show you, and you can look at it on that website Next Strain, where you can look at the viral evolution. From that, you can calculate the rate of mutation of the virus, and it's relatively slow. It's about 25 or 24 24 base pairs per year compared to thousands of base pairs for like influenza. Most of the polymorphisms are just randomly distributed throughout. So this argues that we're not seeing in the current epidemic a lot of viral evolution. That's what you're asking me. Is the virus getting worse in us as it spreads around us? And the answer is probably not yet. It takes longer for this virus to evolve than just the months of epidemic that we've been suffering through. Good question. I got it. Took me a while to understand it.
Great. Lewis, you have a question.
Yeah. So Cliff, that was great. What I was wondering is if you just look at children and stimulate their cells, even in a plastic dish, like through TLRs, do they make higher type 1 interferon responses than do adults?
I spotted some old studies on that using peripheral blood mononuclear cells and stuff like that, and saying that cells from older patients or older subjects tended to make less robust, both NF-kappa B driven and type 1 interferon type responses. I don't know what to make a lot of that. Whether or not that's really true. I do think this idea that the presence of ongoing chronic inflammation in older individuals may lead to epigenetic modifications of innate ISGs or other inflammatory mediators to try and modulate the amount of inflammation that we suffer. That may be real. But there's all these great studies that are coming from Stanford. The surveying of the human immunome, and really argue against the fact, other than oligoclonality a little bit, that people that are older have a worse immune system. If you look at many of those studies, so certainly somebody who has a terrible lungs, some structural problem from smoking, of course they're going to be more susceptible. They're more susceptible to anything, but how much this is really an innate part of our immune systems, I don't know. We'll see. I'm waffling.
Great, thanks. Bill, you had a question.
So, Cliff, first of all, fabulous. I really appreciate it. Just one thing on the ACE2 inhibitors. I'm no expert in this, but my ACE inhibitors. My understanding is that yes, giving the ACE inhibitors raises ACE2, which you would think would be bad.
Yeah, I know.
But in fact, in studies in animals, that protects mice. If you delete ACE2 from mice, they do worse. And so there's now actually a trial of losartan, an ACE inhibitor, going on in patients with COVID-19.
Perfect.
Max, you have a question.
Yeah. What's any reading on IL-6 at this level of the innate immune response? This idea that IL-6 has a kind of a putative role in innate immunity that might increase susceptibility. You come across anything like that in all this?
Not really. I think everybody is using it as a marker of an innate response, obviously. Right. And there are trials, I believe, the clinical people can correct me on the call, of blocking IL-6 because there are obviously receptor antagonists that are out there, antibodies that are out there. but
it's in the sort of the compassionate use area at the moment. I think even the
yeah,
fast tracking some of the antibodies.
I'm sure we'll see real clinical trials of that.
Yeah.
But whether or not it's actually exacerbating innate responses that would facilitate viral replication, I'm not really sure. I think everybody is just using it as an inflammatory marker.
Yeah. Okay.
Great. Jason.
Yeah, it's really great, Cliff. The vaccine sort of topic in terms of the likelihood, given, as you say, the possibilities of antibody-dependent enhancement and maybe which is promoting infection by the virus, and also Fc signaling promoting inflammatory cytokine production, it is a concern. But I think when one looks through the sort of composite studies over the last 20 years on SARS-type viruses, it looks promising that the weighting comes down on the side of protection. So I think there's roughly 100 vaccine trials or vaccines in development around the world. I think we can be optimistic. The one I think to point to is the chicken infectious bronchitis virus, or IBV. That's a coronavirus where there is a successful vaccine in use. So I don't know if you had a different read to that, but I just wanted to hope we could stay on an optimistic note in that regard.
No, yeah, no, I agree. It is possible to modulate the ADE response by altering the adjuvants with which these vaccines are given. So if you give an adjuvant that really promotes much more Th1 immunity rather than something like alum, you tend to get a different AD or a diminished ADE sort of outcome. So even perhaps with inactivated viruses or subunit vaccines, if the adjuvant is right, we'll get a good antibody response from it. Of course, all those vaccines are going to produce primarily, humoral immunity, and obviously we need probably both cellular and humoral immunity to promote efficacious total immunity. So I'll bet that in the long run, the vaccine is going to be some sort of inactivated virus or something like that. But Jason is absolutely right, and we all know it. There must be a 20 or 30 trials ongoing now throughout the world. Some of them are DNA based. Some are using various inactivated viruses or other things. We will have a vaccine to this virus. What we won't have is a vaccine to the next virus, but we'll cross that bridge when we come to it.
Mark.
Great job, Cliff. That was excellent. I want to follow up on the IL-6 thing. There are several trials open, and NIH has invested a lot in this. In China, they've started to make it available as something maybe to add to the protocols for treatment late in disease. You brought it up in the presentation that maybe late in disease, it's anti-inflammatory, immune modulators that we should be going after. There's definitely concerns about the risk of using them, of course, but I wonder what more you know about what directions might be best in this kind of disease. I know in sepsis, writ large, and ARDS, it's been hard to get a signal for these things actually helping, but maybe focused on one infection, we could find something that would actually make an impact on people who are, maybe nearing the end.
So that's a crystal ball question there, Mark.
Well, but IL-6 is really highly expressed, and it's where all the focus has been. Are we missing something?
Yeah, so if you ask a barber if you need a haircut, the answer is yes. If you ask me what cell type we should be going after, it's neutrophils. I think that if we could modulate neutrophil function early in ARDS, not just COVID-19 ARDS, any kind of ARDS, we're gonna significantly diminish the amount of tissue injury that's due to neutrophil degranulation and other things. But I just said I'm looking through my little lens. there could certainly be other cellular mediators that we could be involved in this. Complement may be another thing that we could think about as a tissue injuring factor in disease. So I think through this experiment of nature, there are going to be a lot of tries for a lot of things, and hopefully we'll find out. I am also, besides a vaccine, I'm also very confident that we will have an antiviral very soon, because the virus has so many viral dependent enzymes like the RNA dependent RNA polymerase, which are just screaming, I'm a therapeutic target. We're already trying various antivirals that have been developed for other HIV and other viral infections. Right away, there's probably 1 billion biotech companies that are doing high throughput screens on some of these polymerases, and we're going to have an antiviral right away. The problem with this infection is that you get infected and you don't know it, and that's so how we're going to use these new tools clinically is thankfully not my job. I'm just a scientist, but somebody's going to have to figure that out. So we're going to have vaccines, we're going to have therapeutic antibodies, we're going to have antivirals, and we're going to have immunomodulators. We're just going to have to figure out how to use them.
Great. So I think this is the last question, which goes back to Max. Max, you there?
No, you muted him.
I got muted.
There you go. Now you're on.
I was just going to say, you said some impression a moment ago that I think is worth actually ending on, which is, you said we will have a vaccine for this, but for the next one, who knows? It's fair to say that immunology and science has had a hard time raising money for future diseases in the past. But this is a kind of time where we could actually start thinking about how we should push forward to get programs to basically predict. If you look at the coat protein, presumably, I don't know, but do you know this? Does it bind the equivalent of ACE2 in bats? So presumably you can do the homology matching to figure out what the susceptibility of some of these things would be if they cross strain, right? And so then you have this ability to say, okay, let's at least get a cadre of reagents together, because these are start to be predictable. There are 6 other CoV-2s out there in bats right now. This starts to be a predictable problem, and it actually is quite interesting scientifically too to see evolution, right? So, I mean, again, there could be something that we could be doing, for the next 10 years that is actually predictive as opposed to reactive.
Yeah, absolutely. I want to remind everybody that the MERS virus, the related pathogenic human coronavirus, uses a different receptor. It does not use ACE2. It uses another cell surface protease. I believe that some of the bat viruses, their particular receptors to which they bind are also significantly different. And bat cells actually switch, those are actually polymorphic genes in bats. So many species of bats avoid infection by having polymorphisms in these receptors. All these receptors have other functions. But you're absolutely right. The more we know about all the zoonotic viruses and the zoonotic ligands to which they bind and immune responses to those, the better off we're going to be in having reagents handy. When another weird animal gets together with a bat in some Chinese market, and oh my God, we're all in trouble.
Steve has raised his hand, so I'm going to call on him. This is the last question.
So, Cliff, what's your opinion of the possible use of aspirin as an immune modulator to prevent ARDS?
I probably, Steve, honestly don't know more about it than you do. There is all this talk about non-steroidals. I think I read something just like yesterday that came from the European Medical Union or Consortium saying there isn't really good data one way or the other, but perhaps we should avoid non-steroidals for patients with fever and pain and use things like Tylenol. I don't know. I don't know. It is the simplest anti-inflammatory though, isn't it?
Great. Well, Cliff, I think we're going to end it there. Thank you again. That was a really fantastic talk. Thanks everybody for joining us. This is the ImmunoX Journal Club. We do it every Thursday morning. Want to always be about SARS-CoV-2, but please join us in the future. Also, I'm noticing that this recording of this is going to be posted to the ImmunoX website as well as the bulletin board, so you can see that on the chat. And I wish everybody, yes.
Adrian, I want to, last thing, I see one of the people on the chat actually chatted out the Zoom link for the Department of Medicine Grand Rounds. That's today at noon. Everybody should look at that. That will complement the science here with some more clinical stuff. I'm looking forward to it.
Great. Well, thanks again, everybody. Thanks again, Cliff. Have a good week. See you next week. Keep safe. Keep well.
Oh yeah, there's the hydroxychloroquine study. Okay.
This is what people
Yeah.
are chatting out to you.
Yeah. Okay, Adrian. Yeah, I'm reading all the chat here. Lots of people. Yeah. Sure. I think you're muted, Adrian. Okay, I'm going to sign off too. We'll see everybody.